In my last lesson, I told you how we collaborated with Shally Mendra's lab to produce a vault that could degrade a family of problematic environmental contaminants, the bisphenols. BPA is the main culprit. Our efforts convinced us that perhaps vaults could serve as a biological treatment agent to clean up contaminated water or soil. Before working on this problem, my lab was trying to use vaults as drugs, employing a manufacturing system called baculovirus that uses insect cells. The insect cell system had already been used to make a vaccine that was approved by the FDA. We could use this system to make milligrams of vaults. We knew how to make milligrams of vaults. If you're wondering, that's what a milligram of vaults looks like. It's 7,700 trillion vaults. But if we wanted to use vaults for environmental remediation, we would need to make pounds of vaults, or even tons of vaults. Hi, I'm Lenny Rome, the Vault Guy. I'm a cell biologist and a nanotechnologist. My passion is a common particle of the human cell called a vault. My daughter-in-law Tara and I are trying to explain what vaults are to non-scientists. Today, I want to cover three things. First, I want to explain codon optimization and why it is important if you want to express a human protein in a yeast cell. Second, I'll tell you what happened when Shally's student Meng expressed the human MVP in yeast. And third, I'll explain why yeast turn out to be a superior system for making vaults. Stick around. You might be surprised at how easy it is to supersize a vault. After Meng successfully made a vault that could degrade BPA, he went after the problem of scaling up vault production. He started by revisiting yeast, an organism that we had previously tried to make vaults in, but we had failed. I'll remind you that yeast are one of the few eukaryotic organisms that do not have vaults. We know this because the yeast genome has been sequenced and they do not have a gene for MVP, so they don't make MVP and assemble the MVP into vaults. Meng tried a fresh approach, including changing yeast strains and optimizing the DNA that can be used for expression. It turns out that yeast and mammals make proteins using the same process of transcription and translation that I explained a while ago. All cells are constantly producing proteins from mRNAs that are made. There are differences in translation between yeast and mammals, and one of those differences has to do with the codons that are used. I'll remind you what a codon is with a bit of a review from way back in episode 7. Of course, if you need a detailed refresher, go back and watch that lesson. I'll wait for you here. So, welcome back. In episode 7, I taught you how DNA can be transcribed or copied into messenger RNA. And this mRNA is actually the directions for making a protein. I explained that DNA and RNA use a language made up of 3-letter words that are spelled out using only 4 different letters. A word is called a codon, and each codon designates specific amino acids to be added to the growing protein chain during protein synthesis. Proteins are built up from 20 different amino acids. Codons provide the key that allows these 2 languages to be translated into each other. I showed you this table of the 64 possible combinations of 3-letter codons that can be made from the 4 nucleotides and their meaning. This is called the genetic code. The genetic code is described as redundant or degenerate because a single amino acid may be coded for by more than one codon. For example, UCU is a codon that designates the amino acid serine. However, serine is also designated by UCC, UCA, and UCG. Now, Here is where codon usage gets important. Because we were using the human MVP gene to make MVP protein, the human gene sequence has been optimized by nature for human cells. Different organisms, like yeast and humans, favor certain codons over others. It is thought that optimal codons help to achieve faster translation rates and higher accuracy. For example, human cells use CUA or CUG most often to encode the amino acid leucine. In yeast cells, however, CUG and CUA are rarely used, and instead the codons CUU and CUC are preferred. The reason for this is probably due to the abundance of certain transfer RNAs, but that gets into fine details of translation that are beyond today's discussion. The important thing to point out is that changing a CUG to a CUC has no effect on the protein produced as both codons designate leucine. To get the MVP protein to be made by a yeast cell, Meng changed the human DNA sequence so that it was optimized for yeast by changing degenerate codons in the human DNA sequence for ones used more often in yeast. The important thing is that although this changes the DNA sequence, this does not change the MVP protein that is ultimately produced. Meng also used a strain of yeast commonly called Pichia that was widely used in biotech industries for protein production. With these two changes, the new yeast strain and codon optimization, Meng tried again to express human MVP in yeast. We were delighted to see that the MVP protein could now be expressed in yeast and that vaults were assembled that looked exactly like human vaults. The vaults were structurally indistinguishable from recombinant vault particles produced in insect cells or in human cells. Meng showed that the vaults made in yeast were able to package and stabilize enzymes such as manganese peroxidase, resulting in improved longevity and catalytic efficiency of the enzyme. His results implied that the yeast system is an economical alternative to insect cells for the production of recombinant vaults. Since yeast can be grown in fermenters, like this small 10-liter fermenter we use at UCLA, or even these 1,000-liter fermenters, this advance meant that if we ever developed vaults for environmental remediation, we could potentially produce pounds or even tons of them using yeast. We later discovered another advantage of using yeast to produce vaults. Yeast could be stably transfected with MVP DNA, meaning the yeast-optimized human MVP sequence can be inserted into the yeast genome and a yeast cell line selected that constantly made empty vaults. In scientific terminology, this is called a constitutively expressing strain. When we expressed an int-tagged cargo in this yeast constitutively expressing cell line, the cells packaged the cargo into vaults in the cytoplasm while the vaults and the cargo were being made. Furthermore, because the concentration of cargo and vaults inside the yeast cell is so high, this packaging is far more efficient than could be achieved by mixing vaults and cargo. Using this system, we produced more vaults, and these vaults were pre-packaged with cargo, thereby reducing the steps and labor needed to purify the vaults. When we compared the packaged vaults made in insect cells to vaults made in yeast, the yeast-made vaults were so packed they appeared to be double stuffed. I'm Lenny Rome, the Vault Guy. Join me in the next episode where I describe Meng's efforts to protect the vault. Something critical to our efforts since, as I mentioned to you before, to some random soil organism, a vault would be considered a tasty hors d'oeuvre. Please remember to subscribe and like. Keep those comments coming. I'm trying to teach you some cell biology, not put you to sleep. If you have a question, it probably means that one of the other 20 people that have watched this lesson could have the same question, but maybe they're too busy or too shy to ask. Please ask your questions. I'll do my best to answer them.